Hey, boys and girls, buffaloes and bunnies. Weather report for September 14th, 2021.
Hey.
Hey. So.
Hello.
What's up? So, where is Dessel Drace?
There's
right
never
here. I mean, I'm right here, Speed. What are you talking about? I'm Dressel.
Yeah. Well, you're as wrong as he is often, but
well, true. I mean, I do believe in quantum consciousness, and that's isn't it?
You finally brought me a paper, the Genetic Environmental Networks for Human Personality, which I posted in the description.
Yay!
Take a look at some point in our lives.
It's going to be a very busy description today.
Yes.
Lots of stuff. Icarus brought us some storm chaser material as well. We
on viruses?
Topdawg. I thought he brought us some. But
yeah, he brought us a podcast on viruses, and then the guy that he's interviewing has a site where he talks about virology.
Cool.
He has lectures, and so I went and looked that up, and I posted that one too. It's for anybody who wants to look into the thing that's killing us and likely to extinct the human race. If you want to know what's going to be the end of us, that's it.
No, nuclear war is going to be the end of us.
I doubt it. It's going to be a virus. Anyway.
What, you mean like Danny?
The reason it's going to be a virus is because humans are too stupid to get vaccinated.
That is a definite sign of the end of the human race.
Yep. That's what's going to get us. Anyway, so Dessel Drace has done 5 papers, at least three or four of which are on junk DNA. And he found this guy named Matic, who is basically somebody who is listed on the wiki as being very interested in destroying the idea of junk DNA. He doesn't like the idea of junk DNA. And he's got like a 2-page paper, not a paper, but an opinion piece, where he pushes back a little bit on some of the junk DNA stuff. And I linked those things in the description, 2 papers, the one for junk DNA and the one opposed to junk DNA. And I'm probably going to link Dessel's reading of the papers. I think I'm going to upload them on the channel. He said I could upload 3 videos because he's going to take them down on his channel, so there's no sense linking to his channel. So, but anyway, one of the pushbacks on junk DNA is, one of the arguments for junk DNA is the onion, which has a 16 billion nucleotide genome. So it's 16 billion instead of our 6. Is it? I don't know if they're counting if that's the haploid genes or diploid. I have to look it up. Anyway, the onion has way too many genes.
But that is, sorry to interrupt you, Speed, but that is interesting, isn't it? Like the onion has more genes than we do, yet it basically does nothing. It just sits there.
It's a very, very simple plant. Now, Standing for Truth says the onion needs those extra genes so that it can survive at the North Pole. But onions can't survive.
I mean, what?
That's what he says. He says so. God put all the genes in the onion so that it could survive in different environments. Well, onions don't even do well in Minneapolis where it's sunny and hot. They do really well in cooler climates.
Well, okay. I have a cool question for you, Speed. Out of out of those 16 billion base pairs, how many of those, how many of actually are actually coding? You know what I mean? How many of them actually produce proteins?
I think it has a similar amount of genes to us, but I'm not sure exactly how many. I don't know the answer to that question. I think it's less than 1%, much less than 1%. Anyway, so now the thing with plants is you can end up with polyploid plants where they copy the chromosomes, whole chromosomes, multiple times, and they have extremely large genomes. And so this Maddock mentions that in his paper and says that that's one of the reasons why people think that there's junk DNA, because it's actually polyploidy. Well, when it comes to the onion, I'm afraid those are not polyploid. It's not polyploid. It's actual. The genome is actually that big, and I haven't looked at the onion genome. But then the interesting thing is the garlic genome is a lot smaller, but the onion is really big. So go figure. The garlic is as complex as an onion and very similar. They're both Allium, I think, is the genus. So, yeah, the onion pretty much says, yeah, junk DNA is really a thing. So, anyway.
Does SFT remind you of like a pigeon? You know, the pigeon bobs his head around and walks around and then knocks down all the pieces on the chessboard and says, I win, you know, that type of...
Yeah, he does remind me of a pigeon.
Yeah.
A cowardly pigeon.
You're all pigeons.
Yeah.
When you call him out on it, he won't show up.
Yeah, he's so bad. Like, you know, everything is, oh, we refuted that. We destroyed that. Oh, what are you even talking about? And every argument, you know, it's just on and on. Like he just, raises the flag. I win.
I found another article you may be interested in, Speed. I'm not sure. Psychologists are learning what religion has known for years. Do you want me to link it?
No, I don't even want to look at that one.
All right.
Yeah, no, go ahead and link that. That does sound like something I'd be interested in.
Okay. Let me get it.
I'm always interested in psychologists learning something.
Yeah. I'm always interested in you learning something.
Yeah.
There's that. And let me see. I also have one that you would like about having like a lot of books about neuroscience. Let me see.
May also be another factor motivating Grower. Grower. Yeah, this guy has a problem with Grower, who came out against ENCODE. Well, you know, like I said, I reached out to the ENCODE people, and they said they no longer use the word functional. Now, the problem is that the word functional, the problem is with the word functional, because functional can mean a whole lot of different things.
Here it is. Best books on neuroscience of the neuroscience of consciousness.
Oh, give me a link to that.
I will. Just
Are you putting?
I'm putting it in the private chat. There you go. You're welcome.
What do we
Looks
got?
like I'm not so useless.
Lady in there. Okay, what do we got from David? Oh, David, do you have the one, the 3-minute one from Danny?
Yeah, that was. Yeah, I gave that to Midnight. You know, that was the one. You know, if you go to that homepage, there's a few other ones. There's a bunch of other ones on there. There's another one real short one. Yeah, if you just click on that homepage of that channel that video is on, there's a few other ones. Another one of them screaming, I guess. I haven't watched that one, but.
Danny Powder Keg. That's an interesting one.
Yeah. I think that's. Oh, wait a minute. This is a different one.
Is this this is 1 minute long.
Yeah, this one's only 1 minute long.
Yeah.
Well, this is a different one. There's a bunch of them on there.
Are you sure it's a different one?
I think it's a different one that I sent you.
One.
Is it the same one I sent you yesterday? I thought it was 2 minutes long, the one I sent you yesterday. I don't know.
Sure.
There's a whole bunch of them on there. You got to look. You just got to go to the home page, and there's a whole bunch on there.
Wow. Guess what? StreamYard changed the share screen so the share audio is on by default.
Oh, there you go.
Finally.
Nice.
So they do listen to their users. Oh, let's give this a listen.
I haven't listened to this. Although this looks like the same one. I don't know.
It is the one.
Okay.
Let's see. Can you hear it? Oh, no
No,
sound.
it's not playing. It's not playing. Put on play. It's not playing.
Trying to hit the beginning. What the hell is going on?
Human vascular system and
There it goes.
transporting water throughout the tree. I agree with
No,
that.
that's not true. Hearts were responsible for that pressure.
Don't say, Danny. Don't say it.
Look,
Don't
I
say
have
it.
to
Don't
correct
say
him,
it.
I
Don't say
can't-
it.
Okay, first of all, this guy said that the plant vascular system is responsible for the basically pushing the fluid up the tree. And Danny said no, hearts are responsible. What is he?
Well, we don't know the context, so let's.
I know. Okay.
Yeah.
So anyway, but that's not the part we want to listen to anyway.
Look,
Don't
look,
say it.
BB,
Don't say
one
it. Don't
second.
say
No,
it, Danny.
No,
No,
BB,
no,
you
no,
don't
Danny.
understand.
No, Danny.
BB,
Don't
BB,
say it. I
BB.
want him to say it.
No,
No,
BB,
Danny,
you don't understand.
don't say
I
it,
can't
Danny.
keep
I
up.
do understand.
BB,
I know,
I can't
darling.
keep up
Hold
with all
on.
this bullshit, BB.
I know, baby.
Okay,
I want
so
him to
why are
say
you making
it
me
because
listen to
I
this
want to.
bullshit?
Because I want to
Shut
call
up,
him
baby.
out.
BB,
What
you're
is it?
pissing me off.
Why is he
You're pissing me
pissed?
off
What?
because of sugar. The idea is that if I can't keep up with bullshit after bullshit after bullshit claim, I can't address it all. I have to go one by one. If you give him a monologue, I'm not going to be able to address anything.
What I'm trying to do is
Now,
call
if you
him
want
out,
to fucking
babe.
let him monologue, and let him monologue,
I'm not
then you're
trying
not
to let
interested in the
him.
conversation.
I'm not trying to let him monologue, Danny. I'm trying to call him out. That's what I'm trying to let him. That's what I'm trying to get him to do. Because you taught him all these things, and I'm trying to ask him to show that he didn't remember the things that you taught him. That's exactly what I'm trying to do. I don't want him to monologue.
So can I continue?
Anyway. Danny, yeah, that's Danny Unleashed, eh?
Yep.
Yeah.
So where's the 20-minute one?
I think poor Danny, he's like he was just poisoned by I don't know if that's Discord. It looks like Discord. I don't know if that goes back to like the Google Meet or whatever. That's old.
I'd love to see the whole context of that and see what he was, because I swear he said that it was hearts that pumped
Yeah,
fluid
that's
in plants.
what I thought I heard too.
Yeah, I'm like, what the fuck are you talking about, Danny? I don't know what he's talking about. Maybe he was talking about, maybe it was that Fodor thing. And he was, pushing back because he used the wrong example.
Well, that was, probably because that was back when they developed that. That's back when he was buddies. Well, he still is with Jack Enstrike.
Yeah. Yeah, because there's a whole bunch of Jack Enstrike stuff on this website.
Yeah.
So I guess I'll throw that in the description because we actually, if we have it on the show, it's only fair to put it in the description, right? I'll call it Danny Unleashed.
Yeah.
Yeah, we don't want to let this guy off his leash. Anyway, if you go to that site Midnight, there's a bunch more Danny ones. There's like a 20-minute one that wasn't quite as bad, and it was I don't know what he was going on about it, but he was doing the same thing, sit down. So he hasn't really changed up his game very much in the last.
Yep, he needs help.
Yep. Oh, let me see. What do we got here from Midnight? I got to put those links in. Copy. Anyway, what were we talking about? We were talking about junk DNA again. Oh, I was saying, what the fuck?
Okay, now, just to kind of review what I heard Casper, I keep saying Casper, CRISPR say yesterday was that junk DNA has to do with its source or locality on the DNA. Like you have your house organized into different things, but there's a place or two that you put miscellaneous things that may be useful some way in one capacity or maybe even a new capacity. And so that just, gets put in like a junk drawer in your kitchen or something of stuff you're keeping, but you don't know what for or when you're going to use it. But it comes from just a place in the DNA that's not really organized in an understandable way.
Yeah.
Is that accurate?
Yeah, I think so.
The organized part is my interpretation of kind of what he was saying.
Yeah, and it may become useful later on, like he was saying, right? Isn't that what he was kind of saying? You never know, and we don't know that could develop into something. That part, right?
We have a new guy who apparently walked into the wrong channel.
Yeah, or it may just disappear, or not disappear, but whatever.
Oh my God! It's Dressel Drake, the devil incarnate. Speed, you gotta hide him.
The psychopath.
Yes.
Who puts who does 5 videos in one day and then takes them all down.
My reputation really precedes me here.
Yes, it does. Yeah.
Yeah, we were just complaining about that backstage. How we can never get at your videos.
Yes, we are complaining.
Ooh, Atheist Junior's right. He did a pretty interesting video concerning Kent Hovind today. I will prepare that link.
Okay. So anyway, Dessel, so you did that one short paper by Matt Maddock. Did you look him up? He's got a wiki page article on him.
I heard, no, I didn't know about his wiki page.
On the wiki page, they say that he is sort of hell-bent on pushing back on the idea of junk DNA. Now, one of the things he said in the article, did you hear what I was talking about with the onion?
Yep.
He was saying that, one of the reasons why the genomes are larger, the C-value paradox, is because of polyploidy. Well, that's not the case with the onion, apparently.
Another thing he said in that article, though, is that he's cool with the idea of there being junk DNA for the lungfish or the onion or things like that. But what he wants to concentrate on is the amount of junk DNA in humans. Basically, because the dynamics isn't always the same, you might have more junk DNA in one species and less in another.
Yeah.
So is he pushing back for philosophical reasons about humans,
it sounds more like he sounds kind of similar to, and I can't speak very much for him because my experience with him is really only just this article. But the way that he describes his position in the article sounds pretty similar to people like Hahn and Kern, the contemporary selectionists who think that selection actually does play a more robust role than many modern evolutionary biologists and population geneticists would like to say.
Yep.
Now, what definition does he give for junk DNA? What is junk DNA?
I think he would go with the pretty standard definition that are pushed by the junk DNA guys, where ultimately what you're talking about is something that doesn't have any function.
Yes, and I don't like the phrase junk DNA, and I don't particularly like the word function unless you're applying it very specifically.
That one of the papers was by Doolittle, and he talks about understanding proper function in terms of selected effect, and I very much agree with that. There are certain and I'm going to talk with you guys about this today, but this is still something that I need to study a lot more about. I'm a total noob when it comes to this topic. When you start getting into things like population genetics and junk DNA, this is still stuff that I'm learning about. So everything I say today with you guys is just going to be really cautious and tentative because it's not my expertise at all.
Similar. So.
Shit, I lost my last thought there.
Oh, you were talking about Doolittle.
Right. So Doolittle's got selected effect. There's one thing he says that I don't quite like, because when you're talking about the selection versus neutralist thing, one of the arguments that he gives for neutral theory is that you could have a situation where there's a mutation, and that mutation basically works with some other feature of the genotype, such that if it were eliminated, it would have a deleterious effect. It's not something that positive selection called into existence. It's just something that resulted from the random drift of change. But nevertheless, if you got rid of it now, it would have a deleterious effect because of how it's impacting the rest of the genotype. And so he says that because this is negative selection and not positive selection, and it came into existence by virtue of drift, this is better understood from a drift perspective. But I don't really think that flies, because the only reason why it's sticking around is because of negative selection. So he admits that you have to invoke selection in order to understand it, but then he wants to kind of put the selectionists in a position where they have to maintain that it's all a result of positive selection, and I don't really think that that's true.
Yeah, I think the problem is that if suppose you end up with a long insert, long section of DNA that's inserted into the genome, and because of a couple of other changes in that particular genome, it happens to create a viable organism. Okay, so we got a viable organism, and we have this long repeat. Now that is going to go up around the inner surface of the nucleus, and a lot of it is going to get stuck up there, and it's going to end up with heterochromatin, which is not transcribable, right? So, but the length of that thing may end up being important in that it just happens that it folds up in a certain manner so that everything works. So then down the road, when you end up with a population that's sort of dependent on that loop of unused DNA, untranscribed DNA, right? It doesn't transcribe into a protein. It just happens to be that loop. So now you got the problem of, if you take that loop out, you may screw up the epigenetic factors and you won't end up with a viable organism. So I think that's kind of what they're talking about, because it isn't just about what's in the DNA. It's about how long it is and how it loops up and gets folded, and how it gets attached to the inside of the nucleus in these chromosome territories that's going to affect how the entire cell works. Now, the thing is, there's a big mess of shit going on, and you end up simplifying the whole thing to say either the cell lives or the cell dies. Either the cell turns into the tissue type you need, and the organism lives, or it doesn't and the organism dies. So overall, that's what has to happen. Now, you could end up churning it up and making a ton of changes and come up with a viable organism just because things happen to land the way they did, right? So, I mean, it isn't like you change a whole bunch of stuff and you end up with a dead organism. You could end up with a perfectly viable organism. But now later on, if you just cut one part of that out, you kill the organism. So now we have an unviable organism. Do you follow that?
Yup. That kind of touches on, there were two other points I wanted to get out there. It touches on one of them about conservation of the sequence. And that's something that it looks like they talk all about a bunch. And so there's this one argument that's given. I will try to find the paper. It's a very short paper, but it's often quoted, where the person is talking about how some people expect selection to be able to do things that it cannot possibly do, a kind of forward thinking that natural selection is not capable of doing, such that when you guys were talking about before about how junk DNA could be there for like later uses, the way that CRISPR was describing junk as not really being garbage, but potentially useful for stuff, depending on the circumstance.
Right.
But to suggest that junk DNA is there for that reason, seems to go against the actual dynamics about how selection really works.
No, I don't think it's there for that reason. I think it's there just because of the way things, because of the jumbled mess we have on duplication and because of the transposons. That's why we end up with a lot of junk DNA, or we end up with a lot of DNA that doesn't do anything. So the reason it's there is not to promote evolution. The reason it's there, well, there's one little catch here. In organisms with gigantic genomes, they tend to stay the same. Like you'll end up with what they call living fossils because nothing changes them because they have so many duplicated genes. That can happen. But I mean, none of this is on purpose or, needs to be that way. Well,
or
except for introns.
Or purifying selection with respect to them being dependent at to stick with a particular niche or environment, like the coelacanths and all that. But the point that you guys are making there about the junk DNA and how it's useful for future purposes, and some people like that author that I had in mind would object to that, saying that if there's no actual selective pressure in the present, you can't really say that it's there for that selected effect reason. It's not a forward thinker. But I think that there's like a response that you could give to that in the sense that, that paper I posted a while back on the evolution of DNA repair pathways. It was talking about the role of oxygen in the development of certain DNA repair pathways, the great oxygenation event, and how the onset of that led to the development of these repair pathways. If you looked at the repair pathways, you might think in terms of, well, that seems to be like a forward thinking process. But when you get a population into an environment where there are certain factors that aren't lethal, that provide as environmental stimulus, that dynamic of selection, I think, can lead to the emergence of things that would appear or mimic forward thinking processes, like DNA repair pathways for oxygen. So in a sense, like when you take a look at Koonin, for example, he talks about how the DNA code is robust in terms of downplaying mutation, but it's not the most robust. And so if you consider that, where the effect of mutation on functional capacity, if it's not in a situation where it would cause the extinction of the population, but would rather have like a moderate effect, you could expect populations to develop that are more prone to mutation, and so therefore more prone to junk. And those that are less prone to mutation might get weeded out more. So in that sense, you might end up with a situation where you're prone to junk DNA under particular circumstances, given how selection actually works in that setup or in that environment.
Yeah. My pushback on this idea of that requires forward thinking is that nobody thinks that way, or at least nobody should think that way, because that isn't what's going on, and we know that.
Yeah, talking in a straightforward way, there's no, if there's no selective pressure, then it's not going to be selected for or against, depending on the kind of selection that you're talking about. But
yeah, the biggest selection against is when things get balled up and don't work, and you have a dead organism.
The thing that I liked was in one of the papers, I think it was by Doolittle, he was talking about the endosymbiosis with mitochondria and how this gain of energy capacity allows for the development of things like junk. And so, I think that in a really broad way, you could paint that from a kind of from a perspective emphasizing selection, in the sense that if you don't have the energetic capability, it's going to be selected against, which you see in bacteria. Like there's this paper that I just read, the Black Queen hypothesis, where there's a favoritism toward reductive evolution in bacteria because of things like horizontal gene transfer, where they can keep certain things in a community, but don't necessarily have to have that feature themselves.
Right. Yep. And bacteria are quite a bit different than.
Hey, I put a link to Atheist Junior's latest video in the private chat in case you want to put it in the
And
description speed.
okay, and I'm looking at my PayPal account to see if he's paid me.
Yeah, he really needs to, put a little in there.
Pony up.
There is one last thing I wanted to say on conservation. So I was looking at Lauren Williams's account of the evolution of the ribosome, and he has it developing in 6 distinct phases. He says that the distinctions of drawing it into 6 phases is somewhat arbitrary, but overall, what he found is strict conserving or strict conservation of structural features, but not genetic sequence conservation. And I think that also gets into Andreas Wagner, his work on basically functionality and genetic sequence differences, that the idea of functional redundancy, that multiple sequences can give you the same function or a function that will work in the environment. That I think could be an answer to some of the concerns about how you don't see strong conservation, because some of that may be due to functional redundancy, where you still are conserving the structure, for example, in the case of the earlier features of the ribosome, but you don't need the same genetic sequence in order to conserve that structure.
Yep. Wilson, you said? Do you know what year that was?
Lauren Williams. Lauren.
William.
Yeah, and I could find you the papers. I think the most important one is 2015.
Let's see if I got it. I do not. Let's see here. Pretty sure I downloaded that paper, but anyway, we got to find a way to organize these damn papers under, probably a really good place to do that would be that Science and Naturalism forum I have, where we could list papers for particular topics so that we're not always hunting for a paper while we're talking about them. And so everybody can look at the papers. And I don't have anything on Williams here. Let's see, maybe 2015, you said?
Yeah.
Take a look here. Williams. I got Whitson. Nope, none there. Maybe it's in uploaded. I got to go down to the W's, which is way down there. Willis. No Williams. Damn it. Yeah, I haven't named the paper if I have it yet. I haven't renamed it properly. So I can't find the damn thing. Unfortunately. Williams. Let's see. Okay. That was the one on oxygen, right? Or the 6 phases of the ribosome. Which was that one?
That's, I believe the paper is called The Evolution of the Ribosome, but I could be mistaken. The DNA repair pathway thing is totally different paper. If you don't have that one anymore, I'll send you a link to it again.
I normally download everything, so I have them. It's just that they're not. They're often named Life-05-34 something.
Yeah, no,
blah blah.
It's possible that you missed it because I posted it in the live chat one day, and Leo did bring it to your guys' attention after I posted it. But I mean, you were busy doing the show, so I'm not sure whether or not you saved it.
Get one in check poll. Emergence of specific RNA because this scanner. Nope. I don't see anything with Williams in it. Where do you put it? Did you put it somewhere?
Oh, are you asking me whether or not I uploaded it there? I missed that question.
Yeah.
No, I haven't uploaded. Besides like literally one paper, just trying it out. I haven't put anything in your archive.
Okay, we got it. Yeah. And the other thing is you do have access to that ABio folder on the Abiogenesis folder and the papers folder on the Horseman.
Yep.
Google Drive. We should put everything up there. and then we can easily find it. So, but we got to name the papers. We got to name them with the first author on the paper and the year, followed by the year with no spaces, then a dash. Then I like to put the paper name after that. So it takes a little bit of renaming, and I've got 2,700 papers that I've got to rename right now, currently in my folders. And until I rename them, I can't put them up on the Horseman site. So I got the problem.
Yeah, have fun.
Yeah, well, have fun, or maybe my friends could help me out once in a while. I don't know.
Heard. Yeah, I usually work for about $30 an hour. So if you can meet that fee, I'm totally available. Full availability.
I usually tell people that say that, fuck you.
Oh, shit.
Normally that's my answer. But
okay.
Yeah. Yep. I can hire Chinese children to do that same work for 33 cents an hour.
See, I knew Speed was a true capitalist in his heart.
I am. I think I actually am. I got to get rid of this cat. This cat, goddamn, how much do you weigh? For Christ's sake, Thor. Stop eating. Okay. I'm trying to get rid of you. Yep, I don't love you anymore. I used to love you, but I don't love you anymore. I'm done petting you. Jesus Christ. Anyway, so what else? Okay. Goddamn. So I really wish this whole idea of junk DNA would go away, and we'd concentrate on function, and we'd understand that function is basically a gradient from 0 to 60. And highly, highly, highly functional things are highly conserved, yet they're not conserved by sequence particularly. They're conserved by what they do, what their function is, right? So you'll find a protein in a human, and it'll only be 52% identical to the same protein in a mouse, and yet they will do exactly the same job. So that parts of the protein are not going to be conserved. Parts of it are going to be highly conserved. And if you look across the whole genome, in fact, in NCBI, they have a little graph that you can pull up that shows you, it's got lines about how highly conserved something is. And so you can see a little graph, and it's very, very jagged, and it goes from 0 to 60, basically, 0 to the top. And you can see that conservation even is something that is very difficult to apply in a binary manner. Just like junk DNA puts us in a position of a very binary kind of thinking, which is just bullshit. And if it weren't for the creationists, I don't think we'd still be talking about it. And the ideas, I think it would have gone away. And we just talk about functionality and what is functional and what isn't. So anyway, so I downloaded all 5 of your current papers that you have up on your site. Can I upload them all on my site and put them under a playlist?
You could do, like I said in the email, you could do 3 of them if you want to, as long as the selectionist one is included, because I want to keep it balanced.
Why you limited me to 3? I think I know the 3 I want.
Because that capitalist in your heart, it's just too greedy, and you really need to practice empathy and sociality.
Why? Ooh, here we go. Another spam call from somebody trying to hack my phone. Thor, just go away. Oh, God, I got too many things happening. Yeah, I think, you spend the time reading those papers. We should at least, I don't know, I think you should have them, you should donate them to the world. I mean, if you've gone through these papers. Yeah. And if you don't want to put them on your site, you should put them on somebody's site. So, I mean, talk about fucking empathy and social caring. What the fuck?
Well, I'm just trying to get you to be consistent there, Speed.
Yeah. Well, it'd be nice if you were consistent now. You're talking about empathy.
That shit.
Anyway.
Mm-hmm.
Where are they? Let's see. Let me take a look at this bullshit. Okay, so you got 5 papers. Okay, the Black Queen, you got on causal roles by Doolittle. I want that one. I want is junk DNA bunk. I want the extent of functionality, and I want the case for junk DNA. I want 4 of them.
It's almost like, you ever see Veruca Salt in Willy Wonka?
Yeah, that was my hero. That was the hero of the movie, right?
Yeah, pretty much. I play her. I used to have her song on a playlist. I used to play it a lot.
Yep.
Don't care how, I want it now.
Yeah, I think that was the point of the movie, is to be more like her.
Yeah, you're really. Yeah, well, you're doing a good job. You're channeling her pretty well right now.
Did she turn into a blueberry?
She got sent down to the incinerator.
Oh, okay. She was the incinerator. Which one turned into a blueberry?
That's the fat kid.
Okay.
No, that was the greedy girl.
Oh no, you're right. You're right. Yeah, it was the girl.
Yep. She did turn into a blueberry. That was a really good character. That really was a good character. My kind of girl. Anyway. Your point? I want 4, Decil. 4. I want 4.
I want $10 million.
I removed Decil. We got some power. I got some power. Okay, guys, what else we got going? What else are we going to fill up an hour and 15 minutes with?
Junk DNA.
Should just shoot it in the head.
Fill it up with junk.
We could talk. What'd you say that was in the picture of 58 Chevy?
I think it's 56, maybe even a 52. It's a Buick.
Oh, 52 Buick. Okay.
I'm not sure. It's hard to tell. They kind of made them all look like tanks back then.
Well, I know one of our side chat had a memory of one of those cars, and I just wondered if anybody had any 52 Buick stories to tell.
Oh, I used to. That's what I grew up with. And then they came up with the big fin monsters in the 60s.
Yeah.
Early 60s, they had the big fin monsters for 59, 60, 61. They had the fin monsters. They made everything look like it was going to fly away. And then they came up with a more boxy design.
So here's something to throw, and here's something to throw in as filler. A recent paper published in 2021 by Jash, single nucleotide translation without ribosomes.
Ooh.
I am too ignorant to know if I should be impressed or not.
So it's on the origins of translation. There's also a small side piece, a related piece that's like 2 pages long that I also just read and recorded. I'll see if I can find it. That one's actually called The Origins of Translation, and that was published also in 2021. But like I said, that's just like a 2-page paper. This one by Jash is 8 pages long, single nucleotide translation without ribosomes. And the one that's 2 pages long is just called The Origins of Translation.
Okay. Let's see here. Open link in new tab. So Google has decided to quit downloading for me for a minute. It seems like if you download too many things, it gets mad at you. I don't know. Oh, I got it. This took a while. I got to be patient. I got to be more patient. We'll take a look at that one. So this would be a good on science and naturalism. All these should go under evolution of ribosomes. We could put those papers under there, and we could kind of start listing some of these papers. So single nucleotide translation without ribosomes. Oh my God, that is fascinating. How does that happen?
And this dude is, if I remember right, I don't know if it's this dude or the one who wrote Origin of Translation paper, but based out of the University of Albany, SUNY school, which was my neck of the woods when I was growing up.
Cool.
Anyway, I I didn't think anybody up there did anything useful anymore.
Yeah. Me neither. I'll have to look at that one. I'm not even going to bother to put it in the description because you've mentioned too many papers we've already screwed up horribly. Mentioning papers without links is not a good thing. You know what I mean? You did give the titles, and they are findable, I guess, if somebody were to play this back. But anyway, I think I've lost my will to live.
Just now?
Just now. Just in the last 15 minutes. Yeah. Yeah. Yeah, I'm gonna have to go looking for it. Too many things. Oh, what do we got here? I really like the Palazzo paper. That was pretty goddamn good. Don't you think?
Yep, Palazzo paper was good. I also like the causal roles and selected effect paper by Doolittle. I thought that was well argued. There's some stuff, like I said, that I disagree with, like his whole thing about negative selection and neutrality. But the rest of the paper, though, is well argued. Somebody like me who came studying this from a much more classically Darwinian perspective, after reading a lot of the junk DNA material, I can't sit here saying that I don't find a lot of what they say persuasive about junk DNA. They're definitely, in any plausible selectionist account, it would have to admit that there's at least a substantive amount of junk DNA.
I didn't find Maddox's paper to be convincing at all. I thought he was doing a lot of hand waving.
I want to get more into, he listed something like 8 or 9 papers that he's done in terms of the non-coding portions having many different kinds of function, aside from coding. But he makes a case in a bunch of those different articles that there's a lot more going on in the genome in terms of function. So I don't know anything in terms of that case, but I definitely want to at least look into it.
Yeah, for one thing, like in the introns, okay, splicing is complex and has some regulatory effect, and I think Palazzo mentioned a few of those things. But another thing is sometimes it'll read in the reverse direction and find an mRNA in the intron, and there could be a microRNA there that ends up being used for regulation of genes, and you could end up with a promoter or some sort of promoter, and it'll read, it'll create a mRNA in the reverse direction, and that'll do something and have some effect. And it's just all over the place. I mean, you're going to find, the thing is, it's not code. And because it's not code, that means anything can happen. It means a whole lot of analog level regulation is going to happen based on the mix of which particular kinds of sequences get created, right? And it's going to be a mess. There's going to be a mess of them out there. And that's what drives me nuts about this idea that, this is a machine or this is code. It's a big pile of junk, and either the junk works or the junk doesn't, because purifying selection is very finicky that way, right? It's going to pick the things that are viable, and those things are going to end up out there. And if those things have a lot of spurious translation going on, there's a very good chance that you're going to end up with a new biochemical pathway. Because when you look at biochemical pathways, it looks like a spider web with a spider on crack. There are a number of different ways that you can get to one place, and so this whole thing is a mess. And what we're doing every day is that papers are coming out and are finding a new messy piece of this big puddle of mud that is the human cell, and is the living organism. And I call it a puddle of mud. So, but it's exciting because that's where evolution happens too, in this variation.
There's one question I had for CRISPR. I saw that he was in the side chat before. I'm not sure if he's still there. I don't mean to derail what you're talking about here, Speed, but I just want to throw it out there quick. he was talking about selection acting purely on phenotype and it only being an indirect measure of genes. But what about applying selection to selfish genetic elements? Like, how would that work if that's not being expressed phenotypically?
Yeah, I don't think that's a good way to think about it. Okay, what Christopher is trying to make the point that, you know, the gene isn't going to do anything. It has to go through that phenotype function, and that phenotype is always going to be in an environment. Okay, so you're looking at a function that's a complex of the environment and the developmental steps that the organism takes, right, to become a fully functioning reproducible adult, that sort of thing. So you can't think in terms of, I mean, you can look in population genetics, you could pick a gene and you could say, okay, yeah, we see this thing spreading through a population, and you can ask why. But if you claim that that gene is doing it all by itself, then you miss the point, because no gene is going to work in isolation.
So there is that, but at the same time, you know, when I was reading through these papers, one of the things that was stressed a couple of times was that because there are selfish genetic elements working on their own individual lower levels and not at the level of organismal fitness, that's how you end up with a bunch of, you know, quote unquote junk DNA, in the sense that they're just worried about replicating themselves. They don't give a damn about what's going on the state of the organism. And because of that, you end up with a lot of extra stuff in the genome that doesn't necessarily have to do with phenotypic expression.
Are you talking about transposons?
Things like transposons,
Yeah, and yeah, those could be seen as selfish little devils. And yeah, you can look at okay, I've got a cell in my body, and it is an environment, and there are transposons in there, and they are creating copies of themselves and inserting themselves in the genome. That is a phenotype in an environment. That too has an environment, even if that environment is simply my genome. So you can look at many different levels. That's the fucked up thing about biology, is that there are all these overlapping domains that we have to look at. But
yeah, so I'm just going to throw that out there. Just because I'd like to hear Christopher's answer on that one, just, you know, how he would approach, you know, the way that some researchers have gone about understanding selfish genetic elements and fitness.
The best way to understand selfish genetic elements is not to talk about them, or unless you want to talk about them as an organism per se themselves, that they are their own organism in their own environment, in their own population, because they are. Mm-hmm. So you don't want to going down using these every man terms or everyday terms for to apply to biology becomes problematic almost immediately, because somebody's going to take it up and get the wrong idea.
Yeah, I wouldn't really describe them as an organism per se, but I mean, like, in terms of them being responsible for their replication into the next generation, you can analyze fitness on that level.
Why would you not call them organisms on their own?
Yeah, the only reason why I wouldn't do that is because I have like a multi-component definition of life. So if you're not doing like several different things like metabolism and replication and potentially several others, like maybe compartmentalization, though I don't think so overall. Maybe some kind of compartmentalization would be necessary on some level. And so if you don't have all of those things, you don't have full life, but you could describe it as like quasi-life or proto-life, depending on what's going on, or you would just refer to it as a gene. And you can still measure its fitness in terms of its replicative ability down through the generations.
Yeah, I guess if you insist on having buckets with definitions and clubs that have membership requirements, yeah, then I would have to agree with you. But a virus, you wouldn't call a virus life, right?
I would call a virus at least proto-life or quasi-life because it's able to replicate. So, it has one of the features of life, but would I necessarily consider it fully an organism? Not necessarily. It would also depend on the complexity of the virus and what it does, because there's a lot of diversity amongst viruses.
But what if we didn't have a word for pretend we're aliens. We don't have a word for life. We don't have a word for selfish. We don't have any of those words. All we know is chemistry and molecular biology. Okay. Do you see where we may end up not going down a lot of rabbit holes if we don't have those words? We just have the facts. The facts, man. We have some molecular biology going on here, and that's the only thing that we can describe and that we're interested in. That's why I push back against this bucketization of certain terms.
Yeah, for me, it would come down to if the complaint is that we don't have a fully fleshed out understanding of what we mean by life. For me, the project to take up from there is coming up with such a fully fleshed account. I'm not going to care whether or not it turns out to be somewhat fuzzy. I think that's inevitable because you're dealing with a composite object. You're dealing with something that has multiple subsystems, and therefore there will be certain level of fuzziness in terms of when do you have a fully alive organism versus when you don't. That's going to exist along a spectrum because of those multiple subcomponents that all need to be together at the same time in order for it to be like fully qualified to take that title. And so, in those borderline cases, it might have 2 out of the 3 ones that I just mentioned. And then do you call it life? It's kind of blurry, kind of up in the air.
Yeah. What does it serve to have that classification, life?
It serves, I think it serves in the sense that it identifies exactly what it is that you're trying to account for.
Okay. Some people feel that way. Me being a scientist, I just see life all the way back to rocks. I just don't like the classification at all.
How many of your friends are rocks?
Most of them, unfortunately.
Okay.
They're boring as fuck sometimes. I don't know. Every now and then I get my hammer out and I crack them in half and put them under the microscope because they piss me off. I mean, yeah, my friends sometimes piss me off and I crack them in half. So.
Creation Myths asks if we're still on junk DNA. We can totally return to the paper. The couple of things I added were just asides.
Yeah. The paper I got up.
Yeah, Palazzo. If you had anything further you wanted to put in on that one.
Let's see. Well, I picked out a bunch of stuff. To proclaim the demise of junk DNA on a regular basis without properly addressing some of the fundamental issues that first led to the rise of the concept. He basically calls that premature, right? Let's see. What else we got here? Oh, there was some interesting stuff here, but yeah, I don't know. We don't need to go over in any more detail. You read the paper, the whole paper, right, out loud on your...
Yep.
In fact, various early commenters considered the notion that large swaths... Yeah, he made the point that it is simply not true that potential functions for non-coding DNA were ignored until recently. No one's ever ignored it.
Yeah, I think that's one of the things that the creationists really rely on is that narrative that they just considered non-coding DNA to be equivalent to junk DNA. And as far as I can tell, tracing back through the literature and paying attention to those who support junk DNA today, that's not true.
Yeah, and if we just ignored it, then how did we find the function for all this stuff? If we truly ignored it, we never would have looked, right?
Yep, and you take a look at Larry Moran, and he emphasized, he goes through the history of all the different things that scientists were already aware of, and yet still talking about junk DNA. So I think he's actually a really good person. he's known as being a primary defender, maybe, of junk DNA today. He has a blog called Sandwalk you can go to check out his stuff, but he's pretty good.
Oh, really? He defends junk DNA? Oh.
Larry Moran?
Yeah, he defends it. Okay. Yeah.
Yeah, he defends it.
Yep. Yeah. Are they calling it junk shop or junk? Yeah, CRISPR yesterday came up with junk drawer DNA, and that was cool. Yeah, like CRISPR says, no one ever thought it was all, much less useless. Anyway. But they love that straw man, and they won't shut the fuck up about it. So I went out to Fuzzrana's Reasons to Believe site, and they have an email form. That's the only way you can get a hold of anybody. And I asked to interview him, and they sent me an email back asking me for my channel name, which means they're going to come to this channel and they're going to say, nope. But anyway, I have a request for Fuzzrana to come and talk to us about junk DNA. In particular, I would like to talk to him about this Palazzo paper, because there's a whole lot of stuff in here that counters a whole lot of stuff that he said. And the only thing that, is really understandable is this domestic onion, Allium cepi, is a diploid plant with a haploid genome size of 16 billion base pairs. Now, that's not a polyploid, so they don't have that out. So there's actually 6 billion base pairs in the onion. And in the garlic, I think there's only about 6 billion. So the garlic is somewhat less, but I forget what it is. I'd like to look at those genomes and figure out all what's going on. But there was just, a massive amount of duplication and just all kinds of, God, it's tempting to use the word junk. I know goddamn well that it doesn't take that much to create an onion, over a human. Anyway. How do you create an onion? Onions are really pretty simple plants. They're monocots, right? And they're fairly simple as far as their branching structure and everything. So, there isn't a whole lot of structured onions at all. It would be very interesting to look into the developmental morphogenesis of an onion.
Just to throw out there the other cases that they give that are similar to the onion. In the case of amphibians, there's a wide range of difference. And also the example given by Doolittle a lot is the case of the lungfish, especially in comparison to pufferfish.
Well, here's the table, and this is a logarithmic table. So it shows, let's see, birds, reptiles, flowering plants. Look at the range they have. And then protozoa, massive range for protozoa. And mammals are kind of restricted, and so are birds, are even more so. And lungfishes. Lungfishes are exclusively in the high end range, 100 billion, right? Or close to 100 billion for a genome size. They got some explaining to do if they want to say that every bit of that is absolutely functional and was put there by God, and he had a purpose and he has a reason for it. So they missed the part where the vast majority of DNA failed to negatively affect survivability. Yeah, that's the other thing. In Drosophila, I think, what did they remove? Like 2/3 of the DNA, and they ended up with a viable. But then the creationists will say, yeah, it's fine in the lab, but if you put it out into a real environment, it would die right away because it didn't have all that extra DNA to pop up epigenetically and do something. And there is a grain of truth to that, but only a grain.
I don't know exactly about the fly because I know that Han uses the fly. I think the figure is, but I could be mistaken, something like 50% of the genome is under selective constraint. So I'm not sure about the figure you're using there of 2/3 are able to be removed. I do know, though, that in knockout tests on mice, I think that they've gone up to somewhere around 50% knockout on them.
I echoed. How did I echo? Anyway, that's actually in a lot of genetics textbooks, the part about knocking out Drosophila and knockouts on mice. And then, of course, Standing for Truth talks about if you knock out one gene at a time, as opposed to knocking out 3 or 4 at a time, it has a different effect. You know, there's basically a what is it? Epistasis effect. Who's Yawa? Well, should we let Yawa in the room, not knowing who Yawa is?
Maybe. Just kind of keep your eye on it.
Yeah, you should say who. Okay. Hi, Yawa. Yahweh? Yahweh. Yawa. What's up?
You're on mute. Excuse me. You're on mute, Yahweh.
Yahweh. Yahweh away.
Yeah. There's an issue there.
You think?
Yeah. If they're not willing to talk, then get rid of him. You know why? Because he's on a small little tiny box. And he's waiting. Yeah, he's waiting for you to get into a bigger box so he can probably, do like a porn thing or something.
Yeah, come on to private chat and tell me who you are if you want to get in. That'll help.
I'll be right back.
Okay. Anyway.
Yeah, typical that they don't.
Yeah.
He was going to try something for sure.
Yeah, the being quiet is kind of scary. Boldly go speed, lol. Stephanie. I boldly go where everybody else is afraid to go. No, I'm afraid to go anywhere, actually. Without a mask. I'm afraid to go anywhere without a mask lately.
Your friends here, Brett.
Hey, Brett of Life. Hi.
Yeah.
Brett of Life's there. Hello. That was a nice talk I had with you the other day. Pleasant. I thought you were going to beat me over the head with a Bible, but you didn't. I was getting a little bit afraid, Oh, yeah. We lost Yahweh.
Actually, I wanted to try to reformulate what I was saying before about the genetic code and how selection can actually favor the emergence of junk DNA. Because I don't think I explained it very well before. Basically, like the point that I was trying to make there is that natural selection requires variation to work on. And so if you have a genetic code that's too strict and does not allow for those mutations, that won't do very well in that long term. And so selection could actually be acting in such a way that you do end up promoting lines that are more disposed to mutational variants, which is why Koonin points out that the code is robust to change, but is not as robust as it could be. And because of that, that's how you end up with extra junk in the trunk, at least for eukaryotes, because they're the ones who are able to pay for it with the extra energy capacity they have from the mitochondria. That's really what I was trying to get at there. So it doesn't require forward thinking in the sense that there's no selective pressure. There is, in fact, a selective pressure, but because the pressure isn't fatal to the organism, but merely like a hardship to overcome, just as in the case of like the DNA repair pathways in the ongoing increasing presence of oxygen, because it's not fatal, it can give rise to things that mimic looking like forward thinking. That's basically what I was trying to get at there.
Fuzzarana said basically, and SFT and him were agreeing on if basically there was spurious transcription and translation into proteins that weren't being used, that basically it would be selected against. It would only be selected against if it surpassed some energy barrier where all of a sudden the cell was starving to death because it was doing too much of that, right? Now, one way to look at that is if you have spurious translation into amino acids, what happens is they go into the lysosomes where they're broken down, but that acts as sort of a storage mechanism for amino acids because they're returned into the mix at some point. So it's almost like a storage mechanism. There's another really big, powerful storage system around the calcium ions and having to do with bones. And bones are almost as much for storing calcium as they are for keeping the animal, upright and intact, that sort of thing. But anyway, you could look at that as advantageous, the fact that it creates these spurious proteins that get, stuck in the lysosome. They get tagged and identified and taken out as a storage mechanism, which would be somewhat beneficial. So, but anyway, eukaryotes have plenty of energy. I think we have a surplus of energy. That's why we got to be such bloated mothers, a scourge that took over the earth. Even though we're not that popular, there aren't that many of us compared to the humble protozoa and all the other animals out there. We seem to have really, really done a lot to change the face of the earth.
And you really forget the fungi, whatever.
Yeah.
At your own peril.
Oh, they're humble. They stay underground for the most part. They don't even come out. They're kind of like Dessel with his channel. Are you a fungus?
I feel like one sometimes.
Yeah.
Let's see here. Where the hell is it? I do have one that I will keep up for a short time just because it's. I think it's going to be important. Let me see if I can find it here. But it's Grauer's. The hell is it called? Fuck. 1 second. Rubbish DNA, the functionalist fraction of the human genome. It's like it's almost a 40-page paper. But that's going to be my main project today. After this is getting that recorded, and I think that should be an important one. It's frequently referenced, and I haven't read it yet. So
we have a Kirsten in the room. A Kirsten who wants to debate atheists. How come? Why would you want to debate an atheist?
Yeah, I thought Christians like to cook for people. I'd rather have a Christian cook for an atheist than.
Yes. Yeah, instead of debate an atheist. Yeah. We just want to believe what we believe, Stop picking on us. He left his mic. Mic was bad. Anyway. God, we had a Christian in the room and we let him go.
Yeah. And then none of them are too smart, are they? Wow, look at they can't figure out StreamYard, I guess. No, that's right.
I like what Dr. Dan just said. He says also regulating it super tightly would cost more energy than spurious transcription.
Yep. Yes. Exactly. Yeah, I like the idea of having a whole lot of bits and pieces of proteins and genes laying around. And the other thing that Palazzo pointed out is that the promoters that promote the transcription of a gene are not highly conserved. There's these promoter sequences, these consensus sequences that just have to match up enough in order to get the proteins to attach to the DNA to start the transcription process. You can afford to have a little bit of mutation there, and it's still going to transcribe the gene. Now, at some point, if you have too much mutation, you're going to downregulate that gene, and that's where you'll see a problem, and it will be, and then it enters into the selection mix, that sort of thing. But promoters are a little looser, than, proteins are too, for that matter. I mean, you got an alpha helix, you can do a lot of substitutions before you screw up the fact that it is an alpha helix. Right?
You have an urgent message from CRISPR.
CRISPR message. Forget that.
Oh, never mind. That was for creation myths, but still.
Regulating it enough to stop spurious transcription will stop all transcription. Yeah. Yep.
And yes, Rebecca, please come in if you want to.
Yeah. I think I put the link out in chat, didn't I? Yeah, because I'm pretty much we kind of beat this junk DNA into a pulp. Though I would like to continue to beat it on that forum that Topdawg started. Rebecca. Hi.
Hello, Rebecca.
Guys, how are you?
Hello.
Okay, you
Good,
guys
good.
off camera. I'm going off camera. There we go.
Okay.
All right.
Yeah, we don't want people to see us.
Okay, cool. I don't either.
Because us atheists will have spiky teeth like vampires. I don't know if you knew that.
Oh,
Anyway.
you were on my channel the other day. I didn't see spiky teeth.
Yeah, well,
I just have low self-esteem, Rebecca.
yeah, that was in the morning though. They don't come out until the afternoon or the early dawn. That's when the teeth. I mean, if you look at my picture there, I've got spiky teeth.
Yeah.
That's where we are. So
Well,
we're talking about
just
junk.
go ahead and sink those spiky teeth into me, because come on, I'm a young Earth creationist, and you guys really can't stand our ideas.
Why
No,
would
we
you want to
can't.
do that to yourself?
Oh,
And by the way, I can only stay for about 15 minutes, so
okay.
you got
Why,
15
why?
minutes to tear me up.
Yeah, well, yeah. The problem with young Earth creationism, one of the biggest problems is geology, which is not my field, but I've been looking into it a little bit, and I've been looking into like what's going on in Washington, and when you look at Washington and you listen to the geologists like Nick Zentner talk about it, they're talking about going all the way back to I think 1.5 billion years ago. Some of the formations came up 1.5 billion years ago. And he had an example of some limestone that had been deposited. And it was deposited in I think it was like a couple of kilometers thick.
Mm-hmm.
And now limestone deposits very, very slowly. Or it was it was some kind of a carbonate rock. And I cannot see how that could possibly happen in 6,000 years. That's one big problem with young Earth creationism. And then you look at it, and there's these volcanic upthrusts which they've measured to be.
Excuse me, Speed. It's not a matter of 6,000 years. Like if you're thinking, well, I don't know what you're imagining the process for a YEC person would be, but wouldn't it be more like 4,000 years? And
no, it would be like the year.
It would be within a year.
Yeah, the year of the flood.
Yeah.
Basically, all that upheaval happened during the flood. And then in the middle of all that stuff in Washington, there's all these different volcanic upthrusts where they have one caldera and they'll measure it as being 65 million years old. And then 20 million years later, they'll have another caldera that comes up and destroys half of that one and overlaps it. And they've mapped this stuff out in incredible detail, these geologists. And they've gone through and they checked the zircon crystals and a number of other dating methods. And they put together a pretty massive story about how Washington got created. And if you start reading about that, I don't understand how you could hang on to the idea of 6,000 years. And why would you want to, Rebecca? What do you care?
Well, I've kind of learned over the years to, like, when I read something, I question where they get the information. For example, you just said this, you were reading it about how it took 1 billion years for this thing to come up. And so I would start by saying, where do they get 1 billion years? Okay. And so, like, I just don't, like, the scientific community has lost a bit of quite a lot of credibility with me. Not in every area. There's some areas that they have quite a bit of credibility with me, but when it comes to dates, they've lost credibility because of their selective looking at evidence, and also because of the stories they make up to go along with whatever their hypothesis is.
Okay. I don't think it's so much with geology. It isn't so much stories as they've actually done the measurements, and they have all kinds of techniques for dating rocks that don't have anything to do with. Some have to do with fossils, but mostly they don't. And they've gotten finer. Like in the last 20 years, they've gotten it down to like a 5 million year window. down to less than 10,000 years that they've been able to date things. So they're pretty sure of their measurements with this. And I think it's zircon crystals, and I think they have a few other dating means of dating things where they checked it out. And I think you need to look a little deeper into that and listen less to some of the rhetoric that's being promoted by people who have sent in 50 million year old samples to be carbon-14 dated. You can't do that. For one thing, if you do that, you're outside of the range of error. The error ranges, there's only a certain, there's, we can get down to a very small amount of material that you can do a valid test with. But if you end up with too little, your test is going to go all over the place because it's not going to produce any tangible results. So,
mhm.
Go ahead.
If a sample is 50 million years old, then if they send it in for carbon-14 testing, there should be no carbon-14 found in that sample, correct?
No, that's not the case. There's very likely going to be carbon-14 in that sample, a very, very small amount of it. That's the way half-lights work. In order to get rid of all of it, that would
after
almost be
50 million years?
Even after 50 million.
I just wanted to throw some stuff out in here really quick. In the case of contamination, too, it might not be a negligible amount if it's being contaminated. If you're, what is the word I'm looking for here?
Yeah,
if your sample is contaminated. You said some stuff earlier, like when you read, you critically evaluate what you read. I have a suspicion, though, that you did apply a great deal of criticism toward the sciences. But when you read the Bible, I have a feeling that you did not provide nearly the level of criticism toward that as you did toward the science that you were reading. I don't think that, like when you say that you trust science in some areas but not in others, I think that what that's really saying is that in those areas where you already agree or are prone to agree with what they're saying, you're okay to go there. But the second that it goes against something that you think is biblically founded, that's where your skepticism kicks into high gear.
Well, what causes you to make that assumption about me?
My personal past experience with most believers that I meet, as well as most skeptics that I meet, it tends to be the way that humans operate.
Yeah, well, as we continue to talk, I hope you'll see that that's not what I'm doing.
Okay.
I do understand that, but I understand that assumption, but I don't believe that's what I'm doing. I think there's areas where scientists are more prone to storytelling. There's some like scientific fields that I would call that are soft science, and they require more assumptions. And in those areas, there's lots of room for huge error.
Okay. I want to give you an example where I don't think that you have storytelling involved. And then I want to kind of turn around and flip this on its head in evaluating how you're doing it with the Bible. So when it comes to the idea of like an old Earth, you have things like lake varves and coral reefs and ice cores. And with things like lake varves, you can tell the changing of the seasons by things like pollen content in particular layers. And you can trace that back. You can count the layers going back. And in the case of things like lake varves and ice cores, you can trace them back for hundreds of thousands of years. And then likewise with coral reef growth, they're slow growers. And in order to account for how much they've been able to spread across the ocean, you need much, much longer than 6,000 years. So I think that when you actually, instead of storytelling, when you measure the rates for coral reef growth, so that's not storytelling, that's actually going to get what the rate seems to be. And then also when you actually can trace the passing of the seasons by reference to the kinds of things that you find in the layers, such as pollen indicating the seasons, that doesn't require storytelling. That's actually giving you evidential clues about how long has actually passed. But in terms of storytelling, and you saying that you didn't, you have applied the same level of criticism toward the Bible, what about all the stories that are in there? What have you done to really try to test the truth of all the different stories that you find in the Bible?
Okay, well, actually, I think, I didn't say anything about the Bible in my statement, and I didn't intend to. My statement was with regard to science, like with regard to how I treat, scientific evidence. And I think I evaluate it fairly. I don't assume that something's wrong just because it doesn't agree with the Bible. So I believe that both science and the Bible are legitimate sources of discovering truth. But my way of evaluating the Bible is very different from evaluating scientific evidence. So I don't evaluate the Bible scientifically.
That's what I'm worried about.
Yeah, the thing is, Rebecca, where do you get the 6,000 year date from?
Well, and first let me say, I don't necessarily hold to the 6,000 year date. I think there's room for more years than that.
Many.
But I do get the idea of a young earth from the Bible in general. But I want to say this, that I hold young earth creationism like lightly. What I mean is, I'm not firmly committed to it. If I was shown evidence that was compelling against it, I would be willing to switch my view. And I think there's lots of, room in the Bible for an old earth. It says in the beginning, God created the heavens and the earth. And then before God starts saying, before the creation that affects earth happens, there could be a long period of time there. So I'm not firmly committed to young earth creationism as the only interpretation of the Bible. And like I said, if I had a good reason to scientifically reject young earth creationism, I would be willing to do that in favor of old earth creationism. But now I've been a young earth creationist for like 20 years. Going on. Maybe it might be slightly less than that. I've become more firmly a believer in it over time. But it's like I said, that is just because I see some scientific evidence for an old earth. I see some scientific evidence for a young earth. And so I like evaluating the evidence, it's quite difficult to come to a conclusion. So I tend to favor what I think is a more plain Bible understanding of the age of the earth.
I just, what you just said about not being necessarily committed, and if you were given evidence, you would reinterpret your understanding of scripture. And Andrew says in chat that I should rephrase Bible to young earth creationism. My personal view is that the most honest reading of Genesis would be a literal one. So I don't think that it would be appropriate to try to interpret long ages into Genesis. Many people do that. There's a long-standing tradition of doing that. But personally speaking, I think that the best interpretation of the Bible would be one that presents you with a young earth.
I mean,
nice. I agree. I'm just saying I'm open to a different interpretation, but I agree with you. It's the best interpretation.
Well, can't you just see it as a story? There's like just a story with an allegory or something.
Well, I very much do see it as a story, but I see it as a true story. I don't see it as a scientific story.
I meant more a fictional story. Like a story just written by people.
I definitely see it as a story written by people. I don't see it as a scientific story. I do see it as a true story. So I believe the basic elements of the story are true. And so I'm not willing to interpret it allegorically. I think that kind of
But if
destroys the overall narrative of the Bible.
but if science if science says the Earth was created one way, and the story says the Earth was created another way, and you take it as fact, did I misunderstand her?
I think what you're doing is you are assuming that the science contradicts the Bible, and I don't believe that the science contradicts the Bible. So I believe, now there are. I will agree that the majority of scientists have accepted this idea of a 4 billion year old Earth and of evolution. And I will say this, I strongly reject evolution. I think it's absolutely unscientific, and I believe that the majority of scientists have become very foolish for embracing it.
I mean, you can do that. That doesn't really change what happened. You know.
What's that?
I said you can. Okay, sorry. I said you can do that. It doesn't really change what happened on Earth these past 4.6 billion years to get us here.
I know, but you're assuming that what the story that they've told you is true.
I don't think so.
No, I don't do that.
Yeah.
I actually look into how it happened, so I understand the mechanism of what basically.
Right,
how
I think he was talking to me,
species
Speed, but okay.
Yeah, we don't care about you.
Yeah, no one does.
I
What-
do.
what
I
the-
want to hear from both of you. Let me hear what
Yeah.
both of you have to say in response to that.
Well, I don't know. I've always been science oriented. I've never believed, which I don't know. I don't know. Could either be seen as a good thing, bad thing, or neutral thing. I don't know. But yeah, I don't know. To me, it just seems like science isn't always right. Okay, but it's not always wrong. Whenever it is wrong, usually usually it takes some time to correct, but it does correct itself. Now you said that I'm assuming that what the story they say is true, but aren't you also assuming that the story the story in the Bible is true?
Yes, I am.
Then
But
both
at
of
the
us are
same
assuming.
time, but I'm not. But I am accepting the Bible is true, and I'm also accepting science as a legitimate path for understanding truth about the world. So I don't at all reject science, but what you said is true, that scientists in science has often got things wrong. And in the case of evolution, I will say strongly, in the case of evolution, scientists are wrong. And now with regard to the age of the Earth, I don't know. I have to admit that I'm not an expert in that. And I'm not an expert in evolution either. But once you realize some, basic things about evolution and the story and how it really contradicts observational science, then
here's the thing.
I don't see how you can hold on to it except with an appeal to authority. And that's what it always comes down to when people talk to me about this. They want to appeal to authority. Oh, well, scientists say. Well, I understand what scientists say, but that doesn't change our observational science.
Right, observation. But
where does observational science conflict with evolution?
And this will
Good
be
question.
the last thing I can say but I would love to come on and have a longer conversation about this. And I appreciate you guys because you know what? You're being really gracious and you're not interrupting me. And I thank you for hearing me out and being polite. And I really would like to talk about this longer, but I do have to go. So
how did you even find this stream anyway?
Well, because Mike came on my channel the other day.
They just had a talk. Yeah.
Oh, I see. So so you're that. Never mind.
Thank you for coming anyway.
I'm
Rebecca.
that lady.
Yeah, that lady. Sorry.
Rebecca, before you leave, on the screen I have this Nick Zentner's playlist for his YouTube channel.
Okay.
And he's got a ton of videos, and they're fun to watch. He's a really nice guy. And there's just a whole lot of stuff on there that does not make sense with the young Earth. So you might want to take a look at some of these. And
I'll tell you what, Mike. I'll watch one for every one you watch that I send you.
oh my God. Oh my God.
Well, okay, hold
Okay.
on. Before you go, I do have one question. In Genesis, in Genesis, it's it says that that the humans were created from the dirt of the earth. Now, how can that be when dirt creates silica? Sorry, when I can't fucking speak. When
we gotta let her go.
fine, whatever.
Yeah, but you know what? Please save it. Let's plan a time. I would love to answer your write down all your questions and you can bombard me. And I appreciate the guy who brought up the pollen and the ice cores and all that. And so I don't want to leave you guys' questions and responses unanswered. I would love to talk about it more. So
Yeah, we'll have you on. We'll have you on. So and
Okay.
anyway, take care.
Thanks,
I know you gotta
guys.
run.
Bye.
Thank you.
Nice to meet you.
You
Bye.
too.
Speed, I was trying to get a question edgewise, okay? Sorry,
Yeah,
sorry,
but
my
you-
stuttering gets in the way.
Yeah, but she was trying to leave mid-app.
Yeah,
so
fair enough, but still.
I don't know if I should do this or not, but we'll try it.
What, shoot in
Why
the
do
head?
you think you shouldn't do it?
I don't know.
Oh, okay.
Oh my god, Danny's here! Hi, Danny!
Because intentional states are mental states, right?
Yeah,
Something
intentional
like-
state.
yeah.
Hey, Danny, you may start thinking about perhaps getting a haircut, dude.
No, Scott,
I'm going
he
for
looks
that crazy philosophy hair.
He looks, he looks adorable. He looks adorable with long hair. He just needs a dress. Okay, he needs,
I
he
just
needs
need
to wear a
brush
dress.
my hair.
But if you're going to have that kind of hair, you need to wear a tweed jacket.
A what jacket?
Check out Chris for case 9.
Yeah, a tweed jacket with a sweater vest,
Oh yeah.
bow tie.
But y'all are just wrapping up, right? I love Rebecca. She's, I think, one of my favorite young creationists.
She is very nice.
She does seem like a sweet lady.
Yeah, nice. Yes.
Just don't let her talk to Smokey.
I don't think that's a problem here.
No, never mind. I know.
House of Reason has a question for you, Danny.
I don't know what he's talking about.
I mean,
Well, how about this? How about this question?
CRISPR
I can't.
Cas9.
I mean, I probably could grow a little bit, but I'm not constituted that way.
I know you're not old enough. You're not old enough.
I'm not old enough.
I love
It's actually
CRISPR.
strictly true.
I love CRISPR's comment when Midnight says, "I just want to say something really quick." I'm instantly filled with doubt. Screw you, man. Okay, I'm trying.
What is
Trying to
Midnight
find the
talking
right
about?
theme song.
What
for
is
Danny.
Midnight talking about? And you do need a theme song.
No, I was reading CRISPR Cas9 comment about me.
Oh, okay.
Here's my list of questions, Danny.
Okay, let me just do them now. Let's do it now.
All right.
Oh, we're back on junk DNA with Daniel Dennett.
I'm gonna write in the private chat what my email is, Danny.
Okay.
So that way you can just send them through whenever you're done with them. I'm just gonna keep annoying you about them as we continue to approach the 15th, so that way you remember at some point to do it, but no rush.
Yep. Oh, Chumbawamba, tub thumping. That might work. Chumbawamba? Why can't I say that? What the fuck? What am I doing wrong?
Why does that term familiar?
Oh, you haven't heard tub thumping? I fall down.
Chumbawamba. Yeah, I don't know where that comes from, but that sounds familiar.
I don't think it's Is it Chumbawamba? I'm not sure. Used to really. It's a '90s alternative band. Really good. Very fun. Anyway, what the hell's what are the lyrics to that? We have to check that out. That might work. But I was thinking something more along the line of the Benny Hill theme, but more aggravating.
Well, why are you doing Benny Hill? Oh, I see. Well, no, see, I already know. I already know. Actually, no, I don't know any theme song for Danny.
One
never
theme
mind.
song we did for somebody that was kind of obnoxious in Discord was like that mariachi, obnoxious Mexican music. You know.
I don't know. Is that Mexican? Okay.
Yeah, it's Mexican. Except just imagine trumpets.
Okay, I got you. I can imagine it now.
Yeah, I'll hunt around.
Why can't we be friends?
I wonder if I played a little bit of Christian Kraken on Germania's site. I wonder if he got a copyright strike for that, because I want that to be my theme song opening up the show.
You know,
Christian
Burr
Kraken.
is never around when you legitimately need him.
I know.
He'll pop in, say something. I'm not going to say inappropriate, but maybe, and then he'll leave, and then we'll have a legitimate thing to discuss with him, and he won't be around.
Yep. Gur is almost as wrong as Dessel.
On what?
Everything. Anyway. Oh, let's see. Okay. Danny's theme song.
What was going on over
Scott. There you go,
Scott. What was going on over at Digital Gnosis? You guys were doing a lot of laughing. It was.
I don't know. They were wasting their time with that Russian Muslim.
Yeah. What kind of dumb stuff was he saying? He sounded.
Stop paying attention.
Yeah.
Tell me about morality, though.
Oh, yeah.
How can you have morality without God, kind of stuff?
Yeah. Oh, okay. Let's see, Angelo joined in.
Yeah, then he kind of joined us, but I think he's getting tired of everyone ganging up on him, so he decided to gang up on the Muslim with us.
Yeah. Oh, okay. Yeah.
The mighty Boss Stones. That's what I was trying to think of.
Yeah, poor Antangelo. He hasn't been doing very good lately. He's really gets pounded pretty bad.
He seems to be getting a little edgier, like a little more aggravated. I think he needs to get off YouTube for a while and calm the hell down.
Yeah. Yeah, you must feel bad for the poor guy.
Nah.
I feel bad for myself. That was a joke.
Yep. We're going to shoot her in the head pretty quick.
Why?
Because I got to go for a walk, and I got to go to the store, and I got to do.
You walk every day?
I mean, I do have to go for a walk, also. But I have a funny story to tell you off air.
I miss a few days here and there, but usually my son walks me. So we live on a beautiful Minnesota river valley, and there's trails down there that's just really nice. Big barges go by and weird shit like that. Giant oak trees fall into the river. Giant oak trees, like 300-year-old oak tree fell in the river the other day. Not while we were watching. We saw it one day, and it had a lightning strike, and it was burnt, and it was kind of cracked. And then we went back a couple days later, and it was in the river, like way out in the river. Anyway, wonderful place to walk. And I collect all kinds of bugs and weeds, and there's all these signs in the park that say you're not supposed to pick any plants. I don't know what they mean. I don't know what they mean by that.
Yeah, we got a park here with a bunch of ducks in it, and they don't want us to chase them or throw things at them. And I thought, what's the fun in that?
Yeah, what do you mean by that? I don't get it.
I don't understand them.
You guys are weird.
You can't take them home for dinner?
What? Yeah, even that. I bet if I took one home for dinner.
I mean, if you're hungry,
my taxpayer money is keeping those things alive. So
Yes.
I figure I'm entitled to at least one duck. They even have a goose out there.
I actually tried that once when I was really drunk.
So,
out of McDonald's.
Ducks at McDonald's?
Yeah, there was a little pond out behind McDonald's, and there was a whole bunch of ducks in, and I was really drunk, and I tried to take a duck home for dinner.
So the Christian was talking about how you can't have morality if you don't believe in God. The thing is, if Danny was mentioning it on on Digital Gnosis channel, the thing is, if you're a little kid and you don't quickly learn morality, your mom slaps the shit out of you or fucking yells at you all the time. So I mean, like, it's hard to get out of being a little kid without acquiring some morality. It seems to me, like I acquired reality fast when my mom came in the room.
Yeah,
You
you'll
know?
get a sense of morality if your parents have a sense of morality,
Yeah.
or your peers when your peers become very important to you.
Yeah, so it's inherited. Yes, I had word from John Peterson 2 days ago. He's fine. He calls me about once a week, and
Is
he's
he
doing
fine?
okay. He is
Is he
fine.
fine?
Yeah. Yep.
Well, then why isn't he online?
Because he's sane.
Well,
he's
I'm not sane.
None of us are.
That's
I
why
was
you're here.
on Saturday night. I was going to do Sonata, and I noticed that Germania had a stream, and I went on there, and all you guys were on there. And I sat in my chair and I thought, you know what? What the fuck is going on? I mean, Saturday night, and you guys are all sitting on a YouTube channel, and I just said nope, and I turned on a good movie and watched that instead. I don't know.
Man, I watched a bunch of good movies on Saturday.
We can get caught up in this YouTube community because it is our community. I think I have more friends now in you guys than I have in real life or whatever outside of it.
Yeah, that's. I mean, I don't think I have very many. I don't think I have any friends on YouTube. Maybe like one or two, but I'm friendly with people sometimes, and I'm not friendly to others. But I don't really consider anyone friends in any kind of real
fine,
thing.
Danny.
But
We're not really friends.
No, I mean yeah, I don't really think I'm friends with anyone except if I say Speed is my friend, I think I mean something different than when I say like I had friends in high school.
Really?
Yeah.
Yeah, it's definitely different. It really is. I agree with you there, Danny. It is definitely different because you never actually stand face to face with anybody.
If I hit financial issues, let's say I had a friend actually from Discord, right? Had financial his dad got diagnosed with cancer. His mom has COVID. He lost his job. He needed money. He asked me because we're friends, right? I guess I think of that. But if I was in that situation, I wouldn't call any of y'all, and not because you wouldn't, because you probably would help me. But I just don't think I would feel inappropriate. I don't know. It would feel like wouldn't it be weird if I called you and asked like, "Hey, I'm in a tough spot. I need money." It just seems so weird to me if I did that.
Ian and Nate, the artist, and I bought a cell phone for somebody who was in need, and we've donated all sorts of, things where people are in need. I don't know.
Well, this charity. I'm not talking about. Look, I've been charitable with people that I'm not friends with. That's happened on YouTube. I've done that, right? But and on Discord. But I'm just saying that there's like this. I mean, like I said, I would probably go on a stream and then say, yeah, my friend Scott or my friend Speed or my friend Lori, David, I would probably say that, but I think I mean a little bit different, or Midnight, right? I would.
You never mentioned me, you dick.
I would just mean slightly something different than the friend that asked me for help because he was in need. I feel like I feel with a friend, I feel like I have obligations to them. I don't think I have any obligations to you guys. I really don't.
Yeah, it depends on how you're on YouTube. I guess if you're on YouTube as a persona rather than yourself, then maybe you wouldn't attach to people. But I get as attached to people I know on YouTube as I do any other human being. I believe you guys are real people.
Yeah, the mind doesn't really give a shit about whether somebody's in.
No, yeah, I'm not making that criticism. I think you have really good friendships on YouTube or on Discord or whatever. That's not what I want to push back against. Like, I'm just saying, like, you and John Peterson seem like friends. He calls you. Y'all have obligations to each other, probably. If he was in trouble, you would feel like some kind of moral obligation to do something. I don't know.
We met on YouTube, so.
Yeah, but I have no problem. It's not the YouTube aspect. I'm just saying that even though I'm friendly with a lot of people on YouTube, I just don't. It feels weird for me to call them friends in the sense that I say like my friends and I had friends in high school.
Okay, guys, I think we're going to shoot it in the head. But first of all, Danny, I have a bone to pick with you.
Who doesn't?
Hit the like button, Danny. Hit the like button.
Okay, I'll hit the like button. Okay, I did it.
Okay, thank you, Danny.
Mm-hmm.
Okay, I verified it. Now we're going to quit while we're ahead.
Wait, what did you think? What did you think was going on?
Nothing. I just wanted you to hit the like button. I wanted to see if I get you to do it.
Of course, yeah. I just don't think about that stuff very often.
I know. Anyway, okay. Everybody else hit the like button too. God, if it worked on Danny, it'll work on everybody. Okay, guys, we're gonna end it for the day. Go in the back room. Okay. And then I think Midnight has a story for us. Is that what I heard?
Yes.
Okay, cool. So goodbye,
It
cruel
better be a
world.
good one, Midnight.
We're out of here.